Our next topic is carbohydrate metabolism during exercise, largely more prolonged submaximal exercise, and we're going to focus on how the muscle uses muscle glycogen and how it takes up and uses glucose from the bloodstream. If you recall from this overview of energy metabolism in skeletal muscle, we're going to focus on glycogen and glucose from both within the muscle and outside, how it's broken down through glycolysis to pyruvate, and that pyruvate is then oxidized in the Krebs cycle or converted to lactate. If we look at how muscle glycogen is utilized during exercise, the major determinants of the rate of glycogen use are the exercise intensity and the exercise duration. And you can see in this graph how with increasing exercise intensity, there's an increase in the rate of muscle glycogen breakdown, and that over time, at a given exercise intensity, the rate of glycogen depletion decreases. As we'll see in the lectures on fatigue, running out of glycogen during prolonged strenuous exercise is often associated with fatigue. If we think about the factors that regulate glycogen breakdown during exercise that might explain primarily the exercise intensity effects, we see that there's local control from factors that increase in the muscle, notably calcium, which you'll recall from muscle contraction, increases in the muscle when the muscle contracts. Also, the increase in inorganic phosphate that occurs when muscles break down ATP is another activator of the enzymes that break down glycogen during exercise. In terms of hormonal control of muscle glycogen breakdown, a major regulator is the increase in circulating adrenaline. And as exercise intensity increases and those plasma adrenaline levels increase, it binds to receptors on the muscle and through a series of steps activates glycogen breakdown in muscle. The higher the exercise intensity, the higher the adrenaline, the greater the muscle glycogen breakdown. We also know that the availability of glycogen within the muscle increases its own breakdown. So if there are increased stores of glycogen, you tend to break them down at a higher rate. There's been some discussion and debate as to whether increasing the availability of glucose in the bloodstream has any influence on glycogen breakdown during exercise. And to date, most of the studies would suggest that it has a fairly modest effect. In contrast, if fatty acid levels are increased in the bloodstream, that has been shown to slow the rate of muscle glycogen breakdown. And there has been some interest in nutritional strategies that serve to increase fatty acid availability with a view to changing the rate of glycogen breakdown. And finally, an increase in muscle temperature that you see when you exercise in the heat is associated with a greater rate of glycogen breakdown. So when athletes exercise in a hot environment, they often rely more heavily on their muscle glycogen stores, and that has implications for the nutritional strategies that they need to adopt under those circumstances. Another important factor which influences glycogen breakdown during exercise is training status. And an important adaptation to training is an increase in the mitochondrial oxidative capacity of the muscle, and that's associated with a lower utilization of glycogen. And you can see that both the aerobic and the so-called anaerobic utilization of glycogen is reduced after exercise training. And this is a major adaptation which is thought to contribute to the increased fatigue resistance that you see in well-trained subjects during prolonged strenuous exercise. If we turn our attention to glucose from the bloodstream as a source of energy for contracting muscle, we see again that both exercise intensity and duration impact on the rate of glucose uptake. You can see in this graph the glucose uptake into contracting leg muscles at 3 exercise intensities. So as there's an increase in intensity, an increase in glucose uptake, and you'll also see that at any given exercise intensity, a progressive increase in glucose uptake. If the exercise extends for several hours, then over time, what one sees is a slow reduction in glucose uptake as the blood glucose levels decline. And this is one of the reasons why ingesting carbohydrate-containing drinks is often used as a strategy by endurance athletes to maintain blood glucose levels and to maintain glucose uptake during prolonged exercise. In terms of the regulation of glucose uptake into muscle, we need to remember that this process occurs by facilitated diffusion. And what that means is that we need a gradient for glucose to move from outside the muscle to inside the muscle, and we need a special transport carrier molecule to help get glucose across the membrane. There are three main sites of regulation for muscle glucose uptake: the supply of glucose, the transport of glucose across the plasma membrane by a protein that we know as GLUT4, and the intracellular metabolism of glucose. During exercise, with a large increase in muscle blood flow, we see increased glucose delivery to the muscle. There's activation of the glucose transport process, and I'll talk about that in a moment. And during exercise, the increase in metabolism through those glycolytic and oxidative pathways that we've spoken about means that glucose is rapidly metabolized and maintains the gradient for glucose to move into the muscle. In the post-exercise period, a very important process is the recovery of muscle glycogen, and the resynthesis of glycogen is the major metabolic fate of glucose that is taken up in the recovery period. And ingesting carbohydrate during recovery is an important way of maximizing the resynthesis of glycogen during recovery. If we look at the regulation of muscle glucose uptake during exercise, we see that those three processes that I've just mentioned are increased. There's an increase in glucose delivery because of the increase in muscle blood flow. The GLUT4 transport protein, which normally resides inside the muscle cell, moves quickly to the plasma membrane where it becomes a functioning glucose transporter. There's been some debate as to whether the activity of this transport protein can be increased during exercise. And at the moment, at least, there's no evidence that that happens. And finally, as I said, if you increase the disposal of glucose through the glycolytic and oxidative pathways, then that will maintain the diffusion gradient into the contracting skeletal muscle. We know that the availability of other substrates will influence glucose uptake. If glycogen levels are high in the muscle, then we tend to see a lower rate of glucose uptake. The availability of glucose in the bloodstream is very important because it sets the arterial glucose concentration for that diffusion gradient. So if glucose levels are low during prolonged exercise in the absence of glucose ingestion, you'll tend to see a decrease in glucose uptake. And if you increase the blood glucose level by ingesting a carbohydrate drink and absorbing the glucose from the gut, you'll see an increase in glucose uptake. The effects of free fatty acids on glucose uptake are a little less clear compared with those on muscle glycogen, with some studies suggesting that increased fatty acid availability will slow glucose uptake. Other studies have seen no effect. An important point to make in relation to glucose uptake during exercise is that it occurs independent of insulin. The processes that are involved in glucose uptake during muscle contractions are slightly different from those involved with insulin stimulation. And it's for this reason that there's a lot of interest in exercise as a way of improving glucose uptake in those diseases like diabetes where insulin doesn't work as well. Another important consideration is if you're a type 1 diabetic and you have to inject yourself with insulin, if that occurs in close proximity to exercise, because the two stimuli are additive, that can often increase the risk of premature hypoglycemia. And that's why exercising type 1 diabetics often take lollies with them to rapidly increase their blood glucose level if they feel that their blood glucose levels are going low, or if they feel what's called hypo. And finally, the adrenaline, circulating adrenaline, which increases with increasing exercise intensity, its well-described effects on muscle glycogen. The effects on glucose uptake are less clear. Given that the translocation of GLUT4 from inside the muscle cell to the sarcolemma is very important in removing the plasma membrane as a barrier for glucose uptake, there's been a lot of interest in the molecular regulation of this GLUT4 transport process. And you can see in this slide a number of molecules and enzymes that have been implicated in this GLUT4 translocation process. Most attention has focused on two pretty fundamental changes in muscle. We spoke about those in the adaptations lecture, and that is the increase in calcium and the change in energy status, which has an impact on a kinase, the AMP-activated protein kinase. So again, local events in the muscle, changes in calcium and changes in the energy levels within the muscle serve to stimulate GLUT4 translocation to facilitate glucose uptake into contracting muscle during exercise. Just as we saw with muscle glycogen utilization after training, we also see a reduction in the reliance on glucose. This study was done using labelled isotopes of glucose, which enable you to measure glucose uptake and also the appearance of the carbon label in the expired breath, so you can measure oxidation. And you can see that both glucose uptake and glucose oxidation are reduced after endurance training. Now, it's very important that the blood glucose concentration be maintained within relatively normal range during exercise, and this is the role of the liver during exercise. And so just as you see an increase in muscle glucose uptake, there's a parallel increase in liver glucose output. And you can see here that the curves for liver glucose output are very similar to those of muscle glucose uptake. Both an increase in exercise intensity and an increase in exercise duration will increase liver glucose output. And you'll note at the lower intensities here, where the exercise duration is extended, that eventually the liver is unable to maintain the same rate of glucose output and it starts to decline. And in the absence of carbohydrate supplementation, it's at this point, if the muscles are continuing to take up glucose, that the plasma glucose levels start to decline. In terms of the regulation of liver glucose output, it's quite complex and there are redundant mechanisms. We see both feed-forward and feedback mechanisms. By feed-forward, we mean activation of the liver in parallel with the onset of exercise. And it's thought that various hormonal changes and perhaps even some of the sympathetic nerves may play a role. And I'll talk about that in a moment. There are changes in the pancreatic hormones during exercise. Insulin levels tend to go down and glucagon levels tend to go up. And it's thought that these two signals play an important role in allowing the liver to increase its glucose output during exercise. Circulating adrenaline can also act on the liver glycogen stores, which are then broken down to liberate glucose. The sympathetic nerves are thought to play a role, although there have been some interesting experiments in patients who have had liver transplants, and therefore the nerves to the liver have been cut. And when they exercise, they're still able to increase their liver glucose output. So I think this demonstrates, if you like, the redundancy. And usually when there are multiple mechanisms controlling a process, it means that that process is quite important. And we often see that in physiology with a number of regulatory control systems. Just like the muscle, if the liver glycogen store is increased, you tend to break down more liver glycogen during exercise and have a higher liver glucose output. And the primary feedback control of liver glucose output is the blood glucose concentration. So if it increases, you tend to reduce the liver glucose output. One of the consequences of ingesting carbohydrate during prolonged exercise is that you actually blunt the liver glucose output, because the glucose that's being absorbed from the gut can compensate for any reduction in glucose output from the liver. There are two processes that the liver utilises to produce glucose. It can break down glycogen, and this is referred to as glycogenolysis, or it can also take various metabolites produced during exercise, such as lactate, glycerol, and some amino acids, and convert it to glucose in a process known as gluconeogenesis. You can see in the left panel here before training the relative contribution of glycogenolysis and gluconeogenesis to the total liver glucose output. Furthermore, you'll notice that after a period of training, just as we saw a reduction in muscle glucose uptake and oxidation, you can see a parallel reduction in liver glucose output. And interestingly, that reduction occurs in both glycogenolysis and in gluconeogenesis. Glucose from the bloodstream and glucose units derived from muscle glycogen are broken down during a series of reactions that we know as glycolysis to produce pyruvate. This pyruvate has two main fates. It can either be converted to acetyl-CoA and enter the Krebs cycle and be oxidized, or it can be converted to lactate. In terms of the oxidation of pyruvate, the important enzyme which regulates the oxidation of pyruvate and therefore the rate of carbohydrate oxidation is called pyruvate dehydrogenase. And you can see in this slide that again, intensity and duration are major determinants of pyruvate dehydrogenase activity. So as you increase the exercise intensity and you increase the rate of carbohydrate oxidation, that's associated with a greater activation of this critical enzyme. The other fate of pyruvate is conversion to lactate. And a curve that is quite well known is the increase in lactate with exercise of increasing intensity. And you can see in the left panel the exponential rise in lactate as you increase exercise intensity. And this is related in part to the increased rate of glycogen breakdown. There's been a lot of interest in what we term the lactate threshold, or where you get this rather large increase in lactate. I'll come back to that later in the course when we talk about limits to performance, but it is correlated with endurance performance, and so there's been a lot of interest, certainly in the applied sports sciences, about measuring the lactate response to exercise. And you can see that when you exercise below the lactate threshold, there's relatively modest increase in blood lactate. Whereas when you exercise above the lactate threshold, you can see a steady rise in the blood lactate levels. In terms of the regulation of lactate metabolism during exercise, the production of lactate is really determined between the balance between the rate of pyruvate production and the rate of oxidation. The levels of lactate that you see in the blood are in turn determined by how much lactate is being produced by the contracting muscle and how much is being removed from the circulation either by the liver in the process of gluconeogenesis or by the muscles, because it's known that contracting muscles can oxidize lactate. The muscle oxidative capacity will influence the ability of those muscles to utilize oxygen, and the enzymes that convert pyruvate to lactate and lactate back to pyruvate differ in their chemical activity, and the composition of those enzymes will influence whether pyruvate is converted to lactate or vice versa. The supply of oxygen to the contracting muscles is been shown to influence the production of lactate, and there's been much debate about whether that's a direct effect of hypoxia within the muscle or whether there are other changes accompanying low oxygen, such as increase in adrenaline, which will increase glycogen breakdown and lactate. As I said, adrenaline will stimulate glycogen breakdown, increase the rate of pyruvate production, and therefore increase lactate production. And similarly, if you have higher levels of muscle glycogen, because that results in greater muscle glycogen breakdown, you also see increases in lactate. And finally, one of the hallmark metabolic adaptations to endurance training is a reduction in lactate. And you can see here the muscle lactate levels before and after training, with the characteristic reduction in muscle lactate after training. And this is mirrored by changes in the blood lactate, which is also lower after training. The lactate threshold shifts to the right after training, and as I said earlier, this is used in the applied sports sciences as a biomarker of training adaptations within the muscle. In recent years, there's been a lot of interest in proteins which transport lactate, the so-called monocarboxylate transporters, or MCTs. And an important isoform that increases after training is MCT1. And what this does is facilitate the uptake of lactate into muscle and into the mitochondria, and facilitates the oxidation of lactate, which is an important metabolic fate of the lactate that's produced during exercise. So you can see a number of factors influence carbohydrate metabolism during exercise, primarily the intensity and the duration of exercise, and training status has a major impact on glycogen, glucose, and lactate metabolism.